Okay, good afternoon, everyone. I'm Majoni. My research topic is profiling sugar metabolism and accumulation in plums as related to cultivar differences, maturity, and post-harvest storage regimes. A quick background: the organoleptic properties of fruit in general, they depend on accumulated sugars, and in most fruits the amount of total soluble sugars, they change during ripening process where the fruits when the fruits are transformed into palatable products for the consumers. The sugars, they also have health benefits, especially the sorbitol sugar, which is a sugar alcohol that is present in some fruits belonging to the Rosaceae family, especially the plums. They have high content of sorbitol, and this sorbitol is ideal for replacement for sugars for patients suffering from diabetes, cardiovascular diseases, and also other chronic diseases. The sugar accumulation is affected by cultivar differences and also differences in storage regimes and maturity differences also. We need to, because there is limited understanding of sugar dynamics in plum, we seek to understand at which maturity stage out of the 5 stages that are used in this research, that will be used in this research, which one has the highest sugar accumulation, and also for the cultivar differences, the cultivars that are used in this study, there are 12 cultivars which are grown under the same conditions in Stellenbosch orchard. Here we seek to understand how the sugar accumulation patterns and concentrations differ among cultivars with the different ripening behaviors, either climateric or non-climateric, because the sorbitol concentration, they vary between cultivars. So there is need to identify which cultivars have higher concentrations in sorbitol so that we can promote in the health niche market. And also in this study, the storage regimes that were used to simulate the export conditions. And here we also, on the storage conditions, we need to seek understanding on how the storage conditions influence the sugar accumulation, hence the sensory attributes. The specific objectives would be to identify or optimize an analytic technique for sugars in plum fruits and to discriminate the plum cultivar based on sugar composition or concentration, and to investigate the changes in sugars based on the maturity and storage regimes. Just a quick update so far on the cultivar differences. We have managed to answer the question: can we cluster or group the cultivars based on their individual concentrations at harvest? And in this case, we used GCMS to optimize the individual sugars. And yes, they can be clustered into 3 classes, where the first class, where the first class, class 1, it has 6 cultivars: African Delight, Flavor King, Fortune, Green Red, Sapphire, and Staff, and these are associated with high content of sucrose, sorbitol, and total sugars. And class 2, it has 3 cultivars, Angelino, Lamoon, Rubisan, and these ones are intermediate. They are not really high or low, they are intermediate. And the class 3, it has Inba, Letitia, and Sunkiss, and they have higher content of fructose and glucose. And our study, this research, will benefit the breeding programs and post-harvest management practices. I'd like to acknowledge the following. Thank you.